Are we? Who's Hubs SeaWorld Research Institute? So to give you a little bit about our background, we were actually founded by one of the four fraternity brothers from UCLA who were starting to build SeaWorld in the early 60s. They founded our organization a year before the park opened. And so we were founded with a mission to return to the sea some measure of the benefits derived from it. They knew they were going to be taking organisms from the ocean for education purposes, so they wanted to give back. Even before the park opened, even before they had a for-profit going, they started us, and we're a nonprofit 501c3 organization and have been in that capacity since our inception for 61 years now. So SeaWorld's just celebrating their 60th. I think they finished last year, so maybe we're in our 62nd year. But that is Milt Shed right there, one of the founders. And we're named after the Hubs comes from obviously this gentleman here called Carl Hubs. Sorry, his son's Clark Hubs, but Carl Hubs and his wife, well-known scientist, ichthyologist, was at Scripps for a number of years, and so we were renamed Hubs in the mid-70s, dedicated to honor him. So our organization, we have facilities around the country. One of them is just down the road here, about 10 minutes down the road. That's our marine fish hatchery here in Carlsbad, where we do most of the white sea bass work, which you'll get to hear about tonight. Our headquarters are down on Mission Bay. This is the old Atlantis restaurant. If anybody's familiar with that, maybe Mauricio's the only one that would know that here, but that's now converted. When Anheuser-Busch owned SeaWorld, they gave us that facility. That's our headquarters now. We also have two facilities in Florida on the Indian River Lagoon, so down here in Melbourne Beach. So one side you got the Indian River Lagoon, which is a very important ecosystem and habitat, and then across the way is the Atlantic Ocean, where important beaches for nesting turtles. I don't know if I can multitask on here, but thank you. So that's kind of a little bit about our organization and where we're located. We specialize really for the work that we do kind of in what I consider 5 core areas. And one of them is wildlife populations. And that's basically understanding the relationships between marine animals and their habitat, and what they do and health of the ecosystem that they're living in. And so we look for human disturbances, habitat loss and pollution, and effects of climate change on marine mammals. And so that is a whale shark, and we've done a lot of studies globally on whale sharks. So while we're located here, we just don't focus our research here. We are around across the globe doing research. Next core area is kind of our ocean health. This studies, again, marine life and the ocean and how it could be affected by natural or human-induced changes to the ocean environment. So we're studying things like in Florida in the Indian River Lagoon, health issues of the dolphins and animals that hang out in that Indian River Lagoon. What else? Studying, the elephant seals and sea lion populations along the coast here, and especially at the Channel Islands, which are important breeding grounds for these organisms, and how changes have occurred there. Our next area, we call it animal behavior, but it's really about bioacoustics or sound. So how are these animals communicating? How are they producing sounds? Or how are they impacted by human-made sounds in their environment? So, such as, pure pile driving, those loud noises. You've heard about the military, as we try to measure deep ocean temperatures and using these acoustic large noises that they can hear across ocean basins. Well, animals are most likely affected by those as well, too, in their communication and ability to move around ocean basins. Sustainable seafood. This is the program that I've been working on for pretty much my career at Hubs for 37 years. But we're through this program stocking the ocean with white sea bass and other species of fish, which you're going to learn about tonight, as well as learning how to produce seafood sustainably for our country and to feed our country. It's going to be the way that we're going to start producing our seafood here at home. In our lifetimes, we've overfished the oceans. We are now producing more fish to feed the world through aquaculture than we're taking out of the ocean. So the tide has turned, and unfortunately for the U.S., we import 90+% of the seafood that we consume. That costs us about $18 billion. Second biggest trade deficit behind bringing in oil to this country is bringing in seafood. We have the most territorial waters of any country in the world, yet we're doing nothing out there with regards to farming it to feed our people. We're flying all this seafood in, that's costing us money. The carbon footprint from this seafood, to be responsible and sustainable, we need to start doing it at home. So we're working on research that is going to impact and help us be better at growing our own seafood here at home. And then last, education and outreach. We're obviously inspiring the next generation of scientists and educating people on the scientific discoveries that we do. So these are kind of the core areas and where we focus on. So the program that I work on is called the Ocean Resources Enhancement Hatchery Program, or ORHAP for short. So again, here in Carlsbad, and it's really not about a program that we just operate, but we are the contractors to the Department of Fish and Wildlife to operate this program. We've been the sole contractor since this program's inception in the early 1980s. So it's really a joint effort, collaborative effort between ourselves, the Department of Fish and Wildlife, the Coastal Conservation Association, and many volunteers, as you'll see as I move on along in this presentation. So it's not just us. We don't claim to take responsibility for anything that's going on out there, because we know it's an effort, and it's unique. There's no place else in the world, let alone this country, that's doing a program like this that involves so many collaborators and public people and volunteers. And then the hatchery, again, you saw this previous picture. This building is pretty much open on the inside, even though the roof line's kind of broken up there. But that's where all the work happens. Most of the culture work and broodstock are all in this facility. This here is outdoor tanks, raceways we call them. So we actually bring our water in from the lagoon. This is our pump pit. So seawater comes in from the lagoon. They're actually, when this picture was taken, dredging the sandbar, which you can see kind of in this area. And they're getting ready to do this next month. But anyways, we bring in seawater into the lagoon on the average of about 800-900 gallons a minute to go through the facility. And then we're able to discharge out into the lagoon in this area. The water that we're discharging is cleaner than the water that we're bringing in, because it goes through a lot of sand filters and processes before it goes into our tanks. But where I was going, this is all the layout of that building that you saw. So these are all our broodstock tanks. We'll talk about that a little bit. And then this is where the fish spawn. Each one of these systems is on its own temperature control and daylight control. And then we move fish throughout the facility to different systems as they grow and get larger, from eggs to larvae to juveniles, and eventually they move outside. And then we can do experimentation in this part of the facility as well. So why white sea bass? Here's the kind of graphical representation of the landings for both the party boats, the CPFVs, which is in blue, and then the commercial landings, which are the gill netters and the hook and line guys. And these are in thousands of fish. So recreationally, in the 1950s, we were catching over 50,000 fish a year were being landed. But by the time we got here in the early 1980s, we're down to less than 1,000 fish, sea bass, being landed. Commercially, in the red, the same sort of things happened. And so this program was initiated in the early 80s. Our organization was at that time working with striped bass, and so some of the local legislators approached us and said, well, why can't you do this with a marine fish? And so anyways, they got together, created the Ocean Enhancement Program, and that's the stamp you have on the back of your fishing license. When you fish in the ocean waters in Southern California, south of Point Conception, you have to buy that stamp. That money goes to the Department of Fish and Wildlife, and then we get a portion of that as the contractors to operate this program. But the population really hasn't recovered. It's come up a little bit. We thought this little blip right here earlier on, people were going, oh yeah, the program's working, things are coming back. But really what happened was right here in 97 and 98, most likely. I don't know if you recall what happened in 97 and 98. That was one of the strongest El Ninos we've ever had. So warm water coming up from Baja, slug of water, probably pushed fish up into California. A lot of them being caught, but subsequently it's dropped back down. And on average, recreationally, there's only about 5,000 fish. This is, again, in the party boats, bringing about 5,000 fish a year in. And the commercial fishery, again, they were kicked offshore here in the late 90s to fish in federal waters. They can't, in Southern California, fish with gill nets inside 3 miles, which is state waters. So, but they went out, they found fish. I mean, they were catching fish here at the same levels they were back in the late 60s and 70s. But then subsequently it's kind of gone back down again. So there's concern now here, and now we are in the 2020s, that the population is having some problems, and they may need to do some more management. And I'll show you a little bit about some of the stuff that may be coming online. But again, you've got a fish that travels across international borders, and it's not managed internationally, unfortunately, and it needs to be. And so we've got 2 different fisheries going on, but this fish doesn't know to stop at Tijuana. It just keeps going. And so these fish move around quite a bit. They're more what we call a nearshore coastal pelagic fish. And so here's a little bit about what we know about white sea bass. They're in the family called Cyanidae. They're croakers. So most of you know as spear fishermen, they're listen for that croaking. It's only the males that could croak out there. And we know that it's used to synchronize spawning. And all the fish in this family of fish, cyanids, are croakers. So red drum, spotted sea trout, a lot of fish that you go in the Gulf or on the East Coast, they're popular food fishes there well. And cyanids are popular food fish around the world, and they've been overfished around the world. We know that sea bass can go as far north as Alaska. They've been seen off southeast Alaska all the way down to Mag Bay. They can reach about 67 inches and about 93.4 pounds. That was the record fish that was actually speared by Bill Ernst. I think there was another one recently a year ago that was just 1 pound more than this one. But their lifespan, they can live only about 30 years. And I'll show you a little bit about that later here. They're considered a pelagic, coastal pelagic fish. I mean, they roam up and down the coastline. Obviously, their range, southeast Alaska all the way down to Mag Bay here. This is their range, but traditionally they're really seen from about central California down into to Mag Bay, and then there's supposed to be an isolated population, probably genetically distinct, that's up in the upper Gulf. And so that's kind of their range. They're sexually mature at about 3 years of age. They're what's considered multiple spawners. They can spawn multiple times throughout the year. So, you know, as they come up the coast, we believe in the winter months, and start spawning off La Jolla, and then they can move up to, you know, Palos Verdes and spawn, over to Catalina and spawn. So the females can spawn. Usually they're ready to go every in the spawning season, every 10 days to 2 weeks, and then the males are right there with the females ready to go. And the spawning, we know this from spawning events on our tanks, and working with geneticists and looking at the genetics of spawn events that are coming out. We know who females are contributing and what males are contributing. So we know they can spawn, and obviously you hear the old adage about don't put all your eggs in one basket, and that's what they're doing out there. They're moving up and down the coastline, and maybe 1 year conditions are great off San Diego, but the next year they're better off of San Francisco. So the fish in those areas spawn better. So what else? This was the Bill Ernst fish. So obviously he speared that in 2007, I think it was, off of Malibu area. That fish, you'll see a little bit about that. He gave us one of the otoliths out of that fish when we were doing an aging study, and we'll show you a little bit about the ear bone in that one. But anyways, the management, these fish, right now it's you're able to get 3 a day over 28 inches up until March 15th, when it's going to change for those 3 months, and you're only allowed 1 during those 3 months. Commercial fishing cannot occur at that time. And there's been a lot of changes and regulations over the years. So how does our program work? How do we get babies and fish put back in the ocean? Well, it starts with, obviously, you need adults, sexually mature, spawning. So we go out and collect them sometimes ourselves. These are younger sea bass, but they go pretty fast. It'll take another year or 2 in captivity, and they'll be mature and ready to spawn. But primarily, this is over Catalina. So we have groups of people going over fishing for us. We can hold them in these pens, so they'll fish for a couple of months here in June, July. Load up in here, and then we'll hire a boat. This is an albacore boat to bring the broodstock back. We'll bring them back here to Oceanside. We'll come back with our trucks and tanks, load the fish. We always move the sea bass around in slings, especially the adults, simply because sea bass are very sensitive to handling. We can't really, if you do a lot of netting, I mean, in one day, these fish got to go out, get out of the pen, so we got to get them out of the pen, into the boat, then we got to get them out of the boat, into the trailer, and out of the trailer, into the hatchery, so that would be multiple nettings in a day, and they just don't handle that very well. And because they're food fish, sea bass, the work that we're doing, food fish is controlled by the USDA, and so there's really, when we're doing aquaculture and the work that we're doing, we're really limited to one thing that we can use to treat them if they get sick. So we don't want them to get sick, so we're very careful on moving them around, always in these soft vinyl slings. Eventually they go through a quarantine and end up in these broodstock tanks. Now these each individual tanks, each tank has its own temperature and daylight, and we control that in these tanks. So each tank is on its own different season. And so what I wanted to show you before I go on is those pens out at Catalina. Over the last couple of years, we actually, the broodstock that we collected, not only we just collect them and hold them in there, but we did some science and got a paper published on this looking at hooking mortality, because we go out there and we basically do hook and line. And so we're trying to understand how we could be better at catching these fish and keeping them alive. Most of them do, but you get fish that do occasionally die out there. But we've shown that the size of the fish and how deep you're fishing can contribute to, if they get barrel trauma, that means obviously their swim bladder inflating versus not barrel trauma, and also mortality. So this is also size related. Mortality is increasing here. These are actually fish on this figure that are lip hooked, and then these are fish that are caught with a deeper hook. And so it's interesting that when the hook is deeper and the fish is larger, they have a better chance of surviving. But if they're caught in the lip and they're still a large fish, they have a higher chance of not surviving. So that's pretty interesting. But this is all coming from us going out there and collecting our broodstock, just kind of keeping track of it. And in these pens, so these pens are quite large. They're 10 meters by 10 meters, or 33 feet by 33 feet, and about 25 feet deep. So quite large for these fish, and almost simulate, a natural habitat. I mean, it's in a natural habitat, but they're enclosed in this net that's in there. So back to the broodstock in the tanks. So we've got different tanks up here. 1, 2, 3, 4. The yellow line in these figures represents the daylight period, and the blue is the temperature, and that's shown on this axis. And then these red lines represent spawns, and by thousands of eggs. So when you see the temperature and daylight start increasing in the tank, the fish do what they do naturally and spawn. All we do is change that temperature and light. So now we've got same January to December, and all these figures January to December, but now you can see this tank, here in January, March, it's winter conditions. And they're not spawning. However, this tank's in summer conditions, and it's January, February, March, so they're spawning for us. So by staggering these tanks, having different seasons, we can produce eggs year-round. And then take eggs that when we're ready to do a production run, we'll take those eggs, harvest them, and start the production run. So here is us and their eggs in these graduated cylinders. They'll be there all the way. And so in these graduated cylinders, you see this kind of fraction up here is floating fraction. These are fertilized eggs. Eggs that aren't fertilized sink out. Get rid of that bar there. Get rid of that. There we go. Anyways, and so their eggs in 2 days, they hatch out. And we start the process of continuing to grow them up. Eventually, they get live feeds here for the first month, and then we get them on to pelleted feed, which are, can start off small, like fine grains of sand, and eventually as they get bigger, the pellets just get bigger, and that's how we get them to size. So again, they go through multiple systems. Eventually, they end up getting from our tanks inside to a raceways outside, and then from the raceways outside end up going to other systems. We also have, again, the hatchery. We talked about this or showed the pictures. Some of these just experimental systems where we can look at fish nutrition, such as diets and things like that. We also, and I'll talk a little bit about this near the end, about our what's called our IMTA, or Integrated Multi-Trophic Aquaculture. It has to do with our seafood production. Before fish leave the hatchery, though, they get tagged. Every single fish that we release in this program gets a tag put in it. This is the technology that we use. These are different tagging stations. This one only has one tagging, but this is the machine. There's a small fish right there, and there's a needle, and that needle goes into the cheek muscle and injects that small single tag that looks like a piece of mechanical pencil lead into their cheek muscle, and that's it right there. There's the otolith that you see. So I don't know who was looking to cut the head open to figure out where they were. They're right there. Yeah, so that's the secret, right behind the eyes. So right there. So that tag has a unique number on it if you look at it under a microscope. So each batch of fish that we release has its own unique code. So we know if a fish was released when we get it back years later. Could come from Mission Bay, could come from Santa Barbara, could come from Catalina. So we kind of know all that information. We just, when we get it back, we just dissect that tag out. And you think getting the otoliths are to get out. We'll try getting out something like that out of their head. That's a little tricky. So these are the different grow out sites. So the volunteers, this is where I talk about the collaboration. All these sites from San Diego Bay up to Santa Barbara, there's inactive sites, which are the red dots. They're historic sites. Like Santa Barbara was around for about 12, 15 years, and they have since left the program. But Channel Islands Harbor is still around, Marina del Rey, all these white dots, even up at Catalina Harbor. These are places with grow out pens. Most of those are run by volunteers, although we operate the Catalina one and we operate the one at the hatchery down the road here. But all these other programs have their own source of funds to operate their pens and build them, etc. And this is just kind of some of the pictures of the different pens up and down the coastline. So when Santa Barbara was online, they actually had their pen alongside Stearns Wharf, so they were kind of more exposed and they were more of a seasonal one versus Channel Islands Harbor, these places that are in bays. King Harbor was actually doing a land-based system. One of the original sites at Cat Harbor Island's no longer there. This is our pen over here. There's actually 4 of these pens out there. Again, so we'll have juveniles in one pen, we got broodstock in another, and so on. Huntington Harbor. So the different pens, these are all run by, again, volunteers. A lot of these have their own nonprofit organizations as well to generate revenue and monies to help care for and build these pens out. And so they'll get the fish from the hatchery in about 3 or 4 months, so about 3 or 4 inches in size. Hold on to them. The typical, currently how we're operating is we'll get them to them in the fall, and then they overwinter them, like right now, and they're getting ready to release them here starting in March. In the spring, a lot of these grow out sites will be letting their fish go. So that's how we do the grow out sites. We've also have the ability to release directly into the bays, so we've got our transport truck and tanks, and we can sluice them right into the ocean that way. We haven't done this technique for well over 15 years now, direct releases. We try to get fish into the pens because what we found is the time that they spend in there, somehow they're learning, they're getting acclimated to the environment, and their survival seems to be greater, at least to recruit to the fishery, that is to get to 28 inches, than it is by releasing fish directly. So we now try to be more proactive in getting all our fish into these grow-out pens for at least 2 weeks, if it needs to be a short time, but most of the time they're in there for several months. And then this is just kind of a brief history of all the releases that we've done, starting from '88 up into this past year in '24. And you can see the different colors represent the different coastal counties where fish were released. This is the release numbers in the thousands, so obviously the best year we had was here in 2004 with 270,000 fish. Last year it was only about 65,000 fish, and then the black line is the biomass. What's unique about this program, it may not seem like a lot of fish, especially if you're aware of salmon. There are people are releasing millions of salmon a year out there, and same thing with red drum. But when they're releasing salmon and when they're releasing red drum, like in Texas, in these areas, in Texas, they only grow their fish for about 6 weeks, and then they're like this small, and they let them go, and they're in bays. Our fish, when they go out the door, are minimally 8 inches And lately, they've been even getting even larger, up to 10, 12 inches before they go. So that biomass is huge, and probably makes us the larger program of all these places, because the fish we're releasing are quite large. And people go, well, how do you get your fish that large? That could be pretty costly.
We see them.
Well, that's where the volunteers and those pens come in. You know, trying to do that, grow fish like that on land, yes, can get very costly with electricity and pumping and water and all that sort of stuff. So once the fish are out the door, swimming in the ocean, then we try to get those fish back. And how do we do that? And we have several techniques that we've employed over the years. We have our own permits that allow us to catch fish using our small gill nets like this. So we'll go out and try to catch young of the year, the fish that we've just released, especially within the first 2 years after they've been let go and before they recruit into the fishery. So we want to know what's going on. So we've and that's a program that I run for over 20 some odd years. We haven't been in the water though since before COVID. So that's how we do that. And then we actually ask the fishermen to cut the heads off, save them, and we go up and have freezers up and down the coastline where we can go get the heads, bring them back to San Diego here. We'll scan them looking for that little tag. And then we also sample the commercial markets, looking when they come back into the harbors and offload their fish. We're there. You know, it's usually, this happens in June into July, and then we'll scan them looking for tags. So here's what we've seen from those fish that we've released a little bit. These are actually some older figures, but these are juveniles that have only been released, and they've only been at liberty for less than 1 year. And this is kind of, the blue dots here represent release sites, but these red lines represent areas where the fish have gone, at least where they were recovered. We don't know where they may have gone, but in 1 year, it looks like most of these fish are staying pretty close to the coastline. Even releases over Catalina, yeah, they're caught around the island here, but some of them actually cross a very deep channel to get back to the mainland. So we think, you know, maybe there's more important habitat for juvenile sea bass close to shore. However, you may not see that, we don't see that with adults. So here are adult fish, and there's 120 legal size fish caught between 99 and 2008. So here you have these blue dots, the release sites, and then red dots where fish are recovered. So this is the Cortez and Tanner Banks out here. It's 100 miles off the coast. We caught our fish, one from Santa Barbara, another one from Newport Bay, I think after 5 or 7 years. We don't know where they went in that time frame. They could have gone down to Baja a couple times or up to San Francisco, who knows where. We just know, again, where we released them and where we caught them. So it's pretty significant. It seems like a lot of the adults, larger sea bass, are being caught
Richard caught one
offshore islands and these outer banks out here along here, versus the juveniles are just close to shore. Here's a more recent one from 2009 started to 23, where it's 109 legals, and now you see the red dot are the release locations. So these are all the volunteer sites, including Catalina, and the green are where they're being recovered. We're getting a lot of tagged sea bass up here off of Ventura, Santa Barbara area out here. A lot of these have come from the commercial fishery as well. So they're getting them because they're fishing obviously 3 miles out. And these fish are farther out offshore. A lot of recreational ones are caught at the Santa Cruz and San Nicolas Islands here. And then we actually had one fish caught up off of Monterey that was released down here off Carlsbad. So the program really doesn't go north of Point Conception here, but we've had some departments sampling up there looking, and there's probably more of our fish up there. We just don't put a lot of effort up there because most of the money for the program is here in Southern California, so we stay here. But there has been increased landings and catches of sea bass, especially in San Francisco Bay. And so some of the landing operators and the guys that I've talked to up there have saved fish for us, and we've scanned them, but we haven't detected ourselves anything. But we don't really see that many, although we're trying to get more up there. Another way we're trying to do that is just trying to tap into genetics and looking at the genetics of our fish. Are they unique? Can we identify our fish in the wild based on the genetics? So prior to COVID, we formed a collaboration with some of our partners in South Carolina to start looking at the genetics of our fish. So we went through because we have genetic samples in our lab, going back to 1995 when the hatchery was built and opened up. Earlier than that, I forgot to mention we were down in Mission Bay doing the program. But so we sent them all our broodstock samples from 95 all the way through. So they genotyped, were able to all our parents, and then we sent them some samples to genetically look at those, samples of otoliths. So we have ear bones stored, and so they're able to actually get genetic material. They soak these otoliths in a solution and can get genetic material, depending on how clean they are, to look at and see if they could detect our hatchery fish without having a tag in them. And so we sent them a bunch of samples. They worked through COVID, and just about 2 years ago, so we came out with some results that appeared to show that we are detecting more of our fish out there than what we're seeing with our coated wire tags. So we're trying to understand more about, hey, have they lost their tags? Are the tags losing their magnetism? It's just that small, tiny fleck. So we're working on that. We're continuing to work with the folks in South Carolina in the last 2 or 3 years, in 22 and 23 and 24, sending them tissue samples from fish that we're getting from the wild from our sampling to look to see if they can detect hatchery fish that we're not detecting by our scanning. So we're working with them to do that with separate funding, from federal funding for that. So we also, and this is just some of the goals from that study, but the next slide I'll show you. So the reason we got this genetics work started was because we see when we're out there scanning those fish that come from you guys and the recreational and the commercial, we're seeing about 1 in every 300 fish. So that's less than 1%, it's 1/3%. So 1 in every 300, but we're like, wow, that seems awful low. Fishermen seem to think. But genetics is showing, and so we got this started, but the genetics has shown that there probably is more fish out there than. So we want to know, okay, what's going on with that tag? And there's other ways to do it than just genetics. And one of them is stable isotopes. So the chemical composition in that ear bone, in that otolith, when you go to the core, it means that the most smallest piece of that otolith, especially when they're in the hatchery, those first 6 months, they can look at the chemical composition in there and have been able to identify a hatchery fish versus a wild fish by the carbon isotope. So this is the graph, carbon isotope and oxygen, but basically these up here are separated by their carbon, and so these are all hatchery fish. These are wild fish from Mexico and locally here in Southern California bite. So we're able to also, not only genetically, but we can use stable isotopes in the otoliths by sampling and looking and be able to identify hatchery fish that way as well too. So it's multiple ways, multiple tools to skin the cat to understand, are more fish out there than we are detecting with a coated wire tag.
This is bioscience.
Skip over that.
You don't like this.
So the other thing, again, we're still working on, I think we're coming up on our last year of this study, and we're trying to get more genetic fin clips. So last couple of years, we sent out these little fin clip kits. And so if you want to get one of these kits, it includes, I think, 5 vials in it, instructions, and there's some videos, etc. you can see. But we're trying to get more of these clips this year that we can send back to our partners in South Carolina. I mean, last year, it looks like we got about a 14% return rate. In 2024, we sent out 286 clips, or kits, I should say, and 14 were returned, and now they're being sent back to South Carolina, and they're working on those analyses. But we're going to continue one more year here in 2025. So if you want to get a kit, please reach out to us and we'll get them. I didn't bring anything with me tonight. I don't have them assembled, but there's another colleague of mine that's working on this part of the program. But again, we're still getting heads back from the commercial markets and other places. So we're scanning all these still, looking for our tags. What we're doing though is we're saving these fish, these heads now. If we used to scan them and didn't have a tag or detect the tag with a wand, then we would get rid of it. But now what we're doing is we're saving them in the freezer until we get results back from our colleagues in South Carolina to say, yeah, this fish is tagged, but we didn't detect anything with the wand. So we'll go back now and x-ray that fish, that head, and look to see if we can see that tag, like I showed you that picture of that x-ray earlier with a tag in it. So that's what we're doing. And we've actually found a couple that we somehow missed when they came back and said, hey, this is a tagged fish, from the genetics work they did. So, how do you age a white sea bass? Anybody? Just ask them, right? Or you make some sort of croaking sounds. How many times they croak, right? Like, how are you, stamper hoof or whatever? So, this is how we do it. Here's the magic. So, this is the pearl, otolith, sagittal ear bones, what it's actually called. They have a pair of them. All fish have 3 pairs of bones in their heads. So it's not
Three
just
pairs.
sea bass, but the croakers or cyanids usually have the sagittal otolith. Is in this family of fish tends to be heavier calcified. I mean, if you were to take a 300-pound tuna and try to pull out its otoliths, it would be as thin, wafer thin, as this little ruler that's right there. I mean, this is a pretty thick otolith right here. So
Seriously.
every fish is a little different.
I'm taking
So how
it
we
out.
do this is we take that otolith, we embed it in resin. These are like little ice cube trays. We then put it on a low-speed jeweler saw in that resin, because if you try to cut the otolith without being this resin, it's carbonate. So it'll just start breaking up and disintegrate, because we're taking a thin slice, as thin as a piece of paper. And so we'll cut through what we believe is the core, and then we'll look to see, when we make it two halves, which side is looks like to be it's the core, and then we'll take a second sliver. So now you end up with a sliver that is here on a microscope slide, and then we digitize it under a microscope, but we're using polarized light to help bring out bands or patterns in that otolith. So here's what that looks like under polarized light.
Make her dance, right?
And so these two lines are reference lines for us. This is actually the size where these lines kind of end, the variability of what an otolith, how big an otolith is when it's 1 year old. So this fish is obviously much larger than 1 year old. So what do you see? What's the pattern here, up in here?
It's like a tree.
Like a tree ring, exactly. So these patterns of banding patterns of light and dark bands represent like spring and winter types of growth. So what you do is each pair of bands represents 1 year of growth. The light and the dark band, that's 1 year. There's another year, another 2 years. So basically, you go up and then you count along this is the sulcal groove. So this is where the nerves kind of come into the otolith here. But each one of these yellow lines represents a year. This is actually a picture of
So
that,
1, 2, 3,
or the otolith, I should say,
right?
of the Bill Ernst 93.4 pound white sea bass. So if you count those, 24 is what you'll see. So that at 93 and a half pounds, his fish, it was a female, was 24 years old. At 93.4. So they're really considered a fast-growing, short-lived species. And we just got this paper. I just sent it back 2 weeks ago for a revision. After doing a revision, so it's hopefully going to come out finally.
It's good because that's
because no one's done. I mean, people have done ancient growth, but no one of white sea bass, but no one's really looked at sexes.
Bad, like rockfish.
And there's definitely difference in growth between males and females for this species. So the green here represents male growth. And really the oldest male we've seen has been about 16, 17 years old. So they don't live as long as the females. But the female, at about 5 years in age, they start separating growth. The males start slowing down, most likely because they're getting sexually mature sooner than the females, which are continuing to grow and grow and grow. And so usually they're the larger fish that are running around. The dash line is just the composite of combined of both the sexes. So we're seeing different growth in there, and this is kind of important to know this when you're managing a fishery, what's going on and how old these fish are. So again, sea bass don't live that long, probably not more than 30 years realistically. So even you're catching fish that are 50, 60 pounds. You're talking about a fish that's only 9, 10 years old. I mean, people think, oh God, this fish must be old, must be before your program was around, but I can tell you, we've been around for longer than 24 years, 37, 40 years to be exact. So the next record fish could have come from our hatchery. So that's why it's important, for us to get every head back or get heads so we can continue scanning, looking for our fish. The other thing that's important for you to understand and know, because there's going to be some management changes probably in the next year or two for white sea bass. So right now, that they have to be 21 inches, which is right here, 71 centimeters. This is the legal size. So basically, you're looking at a legal fish being roughly around maybe 4 years, less than 4, 3 to 4 years old. That's how long it takes them to get to legal size. Here is when our females sexually mature. When you have a fishery, you have it managed. You never want to have your fishery catching juvenile fish. That is, before they've had a chance to contribute to the population. So here, 71, so this is showing open squares are immature fish. These gray dots are when so these are, each one of these represents a sea bass that has been caught, they've been cut open, they've looked at its gonads inside to see if it's mature or not. And so this curve is showing you what fraction, what percentage. So here, 50% of the fish are sexually mature by the time they're 900. Well, we're catching fish and you're able to spear fish down here. They're not even sexually mature yet. So this just came out in the past year. The department's been working on this for a number of years. So by the time you get up to 100%, you're at a meter, 1,000. So you're over a fish that's over 3.5 feet in length. So they're looking to change this and probably going to be up here. I don't know if they're going to quite push for a meter in length, but they may be here in the 900s or maybe 30. I mean, so that's 43 inches up there. So they may change that at some point, but they're starting some discussions, working with the Fish and Game Commission to change it. I think the commercial fishermen, if they're going to be still continuing to fish, although there's legislation that's going to change the gill netting and whether that still is able to occur here in the next year or two, but they're going to push that they probably can't afford their nets and changing to get something that large. So there may be some compromises here. I'm guessing they're going to be somewhere around they may set the minimum size to around 900 millimeters total length. So that's going to be about 32, 36 inches somewhere in that. I'm guessing what they're going to target. But just to make you aware, this is because of this data. It hasn't been published yet. I mean, we did talk about it in the fishery management back last year, but I think they're working on getting this published. So until it gets reviewed, but this is what they're looking at. So that's it for sea bass, but that's not all we do. And so you're probably interested in halibut. I heard some guys talking about spearing halibut. So we've been working on California halibut for a number of years too, and there's been a lot of interest by the recreational community for us to also be doing what we do with white sea bass, to start doing it for halibut. So we've been working with private funds to actually grow halibut. Now, why halibut? Well, halibut, the same sort of thing is going on here. So they've done some stock assessments looking at the population along the coastline of California here, and they actually separated it. A southern population here in Southern California and a northern population. But what they found is the Southern California population, now this has been over 10 years old, this data, so I think it's even down to less than maybe 10% of the unfished biomass. That means 90% of the fish are gone out there from what we believe is the historical population levels out there. So we're continuing to work on halibut, and it's been a species that has been enhanced with releases around the globe. This family of fish, the flounders, are actually in the flounder family. They're doing Japanese flounder. There's releases over here, the turbot in Europe. We got along the East Coast. You got winter flounder, summer flounder, southern flounder. So all along here in the Gulf, they're doing a lot of work on halibut and releasing flounders, not halibut. And then we call ours a halibut, even though it's not really in the halibut family. It's not in the same family as the Pacific halibut. But we got California halibut down here, and so we're working on there. So we've worked on models, trying to look at, okay, when's the best size to release these fish? You know what occurs in the wild. Obviously, they've got eggs to larvae, juveniles or adults, subadults or adults. So we've done some modeling to suggest a good size, and I forget what the actual size is, but it's more or less at a juvenile size, around a 5 or 6 inch halibut. But halibut don't grow as fast as white sea bass, so it's going to take a little longer for them to get to that size before we can let them go. So we're actually trying to do experiments where we can let them go and try to recapture them and try to learn, because the department may incorporate this fish into the ORHAP program for white sea bass. So we want to be ready to go if they say, yeah, here's some funds, let's go do halibut. So we've collected, we got the same thing with like white sea bass. We got to collect a brood stock. So unfortunately with halibut though, there seems to be some subpopulation structure in it. So no, it's just kind of, there we go. So along the coastline for sea bass, the genetics tend to show us that from Northern California down into Baja, it's one big what they call panmictic population. Halibut, not so much the case, which means they don't move around as much as sea bass. So we know the population off San Diego is probably a little bit different than off of LA and off of Ventura. So at one point we had two populations to work with. Right now we just have the San Diego population. Unfortunately we lost, but we rely again on the anglers that have helped us with the sea bass program are helping us do halibut as well. So same thing, we got to transport them in slings. We'll go out in San Diego Bay or along the coastline, spend the day fishing, catching halibut. It's a little easier to catch halibut than it is to catch sea bass. So it's a nicer trip. We can do this in a day. When we fish for sea bass and we go out to Catalina for 3 or 4 days in a row, you're beat. I mean, it's not a fish that you just can catch anytime. I mean, we're pretty much sleeping when we can catch a few minutes of sleep, but we're constantly going out there trying to catch these broodstocks. So it's definitely a big difference. And you can go down to San Diego Bay and catch some nice halibut and a nice day, nice calm waters, and call it a day. You can go back to your bed. Fortunately, you're out in Catalina. It's a different story. So again, same sort of some sort of way things that we're doing here. And so again, loading in broodstock tanks. You know, we've actually tried to do natural spawning like we do with a sea bass temperature controlled tanks. We've actually tried to inject them with hormones as well too. You can inject them with that. It's pretty hard on the fish. And you end up maybe eventually losing that fish. But we're working on the culture techniques, and this species presents some unique challenges, unlike the sea bass. In particular, culturing these fish, the sex and the pigmentation are really key areas. Sex for this fish can be determined, or is determined, by temperature that you culture them at. It's a slow-growing fish. You know, we try to, hey, let's get the water a little bit warmer and let's grow them a little faster. But what happens is you convert them to males. So water needs to be cold during certain cultures. So we've been working on that. In addition to the pigmentation. So here you see kind of little bit of life history of the halibut and kind of what's unique right here. What's different about this halibut versus this halibut? What's that?
It's got a great hairstyle.
It's got skinnier. Yeah, it's younger by 15 days.
It hasn't shifted.
Smart. Yeah, look at his eyes. So here, when they're still 15 days, they got eyes on both sides of the head. Now the eyes rotated and they're becoming more demersal than they are pelagic up in here. So yeah, so the eyes have migrated here by the first month. So, and then the problem is that look at all these fish here. If they were lying in the sand off the shore here, how easy do you think it would be to see those nice white halibut out there, unless we got some nice pristine Hawaii sand or something like that. But so we need to figure out, we're still working on why the pigmentation, some crops, these are all what we call albinos. I mean, they haven't developed their pigment, they should. So we're working on that. We've kind of got the sex figured out finally, but the next thing is to get that. So here you see a mixture of pigmented and non-pigmented halibut. But halibut are easy to grow, or easier. They don't require as much space as a sea bass. You could grow in these oversized rain gutters, basically. They're just long, narrow. There's only an inch or two of water in there, but here you can see we got thousands of larvae in these tanks, and you can stack them nice and easy. It's pretty hard to stack a big tank with thousands of gallons of water in it. Here you just got a couple inches, so it's really easy to grow them. So it's more practical. So in 2019, we actually did get permission from the department, did a small scale release in Mission Bay in 3 different areas, and we worked with CCA and everyone out there, volunteers. We loaded them up and distributed them throughout the bay in 3 locations, and went out there sampling, trying to recover those halibut. We did catch them the first time, but after the second or third day, we couldn't catch them, so they didn't catch any, so they dispersed pretty well. We couldn't find them. We trawled in a bunch of different areas, and we're using trawls to try to recover these fish. So here's just a paper that we've published here looking at the sex determination. So here's your percent of sex, whether it's 50-50, males, females, or does it get more skewed as it goes up. So here at the different culture treatments, 15 degrees Celsius seems to be the sweet spot for growing California halibut, to get a 50-50. So why is that important? to be putting a 50-50 sex ratio out there. It's important because what's going on in our environment right now with Mother Nature? Things are getting warmer, right? Ocean's getting warmer. Just by nature, there may be fewer females running around out there. So we're trying to put females back into the ocean. So now we know that we need to culture them and concentrate on 15 degrees and making sure our systems do not vary for the first couple of months, keeping them at that 15 degrees. Just cold water. So again, depending on where we're growing them out, that could be pretty expensive to chill that water and keep that down cold enough. So that's a little bit about halibut. Kind of coming towards the end here, wrap this up. But I'll talk a little bit just quickly about, we talked about seafood production and what the other work that we're doing. So this is the multi-trophic aquaculture. So we're starting to get money in this area. The U.S. seems to be favoring this type of research. So basically, in the short, you're using, and we're kind of unique. This fish hatchery down the road in Carlsbad is the only marine fish hatchery on the west coast of the United States. So it gives us a huge advantage in trying to do these type of studies where the fish that we're growing can be used, effluent from those waters and those tanks, can be used to grow other species invertebrates and algae that are also edible seafood. So we're working and gotten funds to do that and build multiple systems. And so we're doing some of this work at our Mission Bay office location here and have built systems. We have California yellowtail down there. That's a system, and so we're designing this where we're putting, here's the fish providing the nutrients to the algae, and we also have sea cucumbers, which are very popular in Japan and very expensive. So anyways, this is, I went backwards here, showing the flow of the water, and this goes through the systems and how we're able to then recycle this water and feed these organisms that are growing at a faster rate. They're very popular to eat for food items, whether it's the sea lettuce or sea cucumbers or mussels. At the hatchery here in Carlsbad, we have this gravity-built system where we put sea bass in these tanks, and then it trickles down just by gravity into these other tanks, of which we have algae, and we can put other invertebrates, and we have sea cucumbers and stuff up there too. So we have permits to collect these organisms, to start looking at growing these things out through this integrated multi-trophic aquaculture. And what's interesting is that some of these algae can also be used to help clean up the environment and the water, especially this species of algae called ova. It's like tissue paper, and certain bayments it can be everywhere and clogging things up, but it's a very, very efficient nutrient remover, scrubber. So here we've got ammonia nitrogen. It's moving, in our studies, 44.5%. You've got nitrate in the water, 65%. You've got nitrite, and you've got phosphates. We get a lot of that stuff coming down when we have rains coming down the canyons and into these oceans. And so ova is a great source for that to clean that up. In addition to, it's an edible one. I don't know if it's one of the more popular ones, but it can be edible and used in seaweed salads and that kind of stuff. So, and lastly, we talked about education as one of our core areas. It's a program that I initiated some 15 years ago now, but called Seabass in the Classroom. There's over a dozen schools in Southern California that have purpose-built systems in their classrooms, just like we have at the hatchery, to grow sea bass in. So we'll bring sea bass into the classroom. They get them in a couple inches. And so different schools have different systems. Some have more money than others and build their own high-end fancy systems. As a matter of fact, this one's even changed. This tank's even bigger now. He's got more bigger systems, and this teacher's just so fired up about this program. And I said, yeah, well, with bigger systems come bigger headaches, but if you want to keep going, knock yourself out. So he's got a lot of money. He's great. I mean, this whole lab that he's got used to be the auto repair shop. That's now the innovation center. So he's doing a lot of different aquaculture projects. He's doing trout in the classroom and all kinds of stuff. But these students have these systems just like we have in the hatchery. I mean, but they can learn about, this is the newer type that we're going to. These are portable because a lot of teachers, when we built them into the classrooms, they have to change classrooms. So it makes it hard to go use this if they're in different classrooms. But these are portable, and they've got everything they have that these systems do as well. So they've got nitrification going on here in the sump. They've got a UV filter. You can't see it. They got a gas stripper. They got a chiller. What else? They got a protein skimmer. So they got all the technology that students can learn about the technology. I mean, they're our next generation of scientists, engineers that are going to support aquaculture here in the United States. So this program, obviously we just talked about what they're learning about. They're learning about agriculture, because aquaculture is agriculture. We're running out of land, we're running out of fresh water, we're running out of resources to do terrestrial aquaculture. And it's not an efficient process to do terrestrial versus fish in the ocean. Just the protein conversion is much higher, better I should say, in the water than it is on land, because you fight gravity, so you're spending a lot of that energy to fight gravity, generate your heat, because you're endothermic, right? Fish are poikilothermic, so they just the temperature of the water, so they don't have to use that energy, so they can devote it more towards growth. So they're learning about that. And so how the program works is we take the fish to the classroom. They do this twice a year, so each semester in the winter and then the fall. So they'll get the fish, they hold them in their classroom, we work with them, we go there and we anesthetize the fish. They get to measure and weigh them before we put them in the tank. They feed them. They can use and do other studies in the classroom while they're doing this, and they can learn about food conversion and that. And then we go back later in the semester. We'll work with the students to tag the fish in the classroom. So here's a student tagging the fish. This is at Mission Bay High School a few years ago. And then they get to go release their fish at the end. So they'll check them for the tags. Here they're checking them before they let them go. And then they just let them go in the various embayments. Some teachers prefer to go out. This is off La Jolla, and they'll kayak, and they put them in the ice chest, and they'll go off and release them off La Jolla shore there. So this is our next generation of scientists right here. So with that, I'll take any questions. But if you have a question to ask me about, have I ever speared a sea bass? The answer is yes. I just did it. There you go. Thank you. Yes.
Mike, thanks so much. That was an amazing talk. And I have a bunch of questions, but I'm going to leave it with just one open question and then a comment. So the open question is, what can we as recreational spear fishermen and women do to help out with the project? Maybe just summarize some points on that, and like what can we do to safeguard the species as much as possible. And then I wanted to say, if you have a handful of those fin clip kits, you wanted to hand them off to some of the club leadership, we can distribute those to members as well.
Yeah, so we'll have to work. I'll get a card. I've got John's email, and so I can get them, figure out how we can get them the kits to you, and I'll